Hey everybody, it's another video tutorial with Dr. Ramey. This tutorial is going to focus on aseptic technique, some basic lab protocols, and how to work with live organisms in the microbiology lab. Okay, so the first thing is my materials. I want to make sure, well actually the first thing is that I'm properly dressed, right? I've got to have the proper attire before I can even begin. All right, so this is one of the reasons why it's a hard and fast rule that you have a proper lab coat. For example, you can see that I'm wearing my lab coat. You're going to have gloves. I'm not going to use gloves today, but some days you're going to be required to use gloves and safety glasses. Okay, at a minimum, you can never work with live organisms without your safety glasses and without your lab coat. All right. So that being said, then I'll make sure that I have all my materials. So I'm going to have a sample here, a culture. Okay, so I'm going to. It's clear right now. It should be cloudy, but I don't have. There's nothing grown in it. But we're going to pretend that this is my broth culture. Now sometimes you might get organisms on a plate, or in a broth culture or on a slant, and we'll talk about what those are in lab or in a future tutorial. But today I'm going to demonstrate just basic aseptic technique. That's what we're going to focus on. Alright, so I'm going to demonstrate the aseptic technique for transferring, or subculturing as it's called, some bacteria into a liquid broth, okay, a nutrient broth, excuse me, and onto a solid medium, which is nutrient agar. Okay, so this is what I refer to as a plate, right? So we've got a nutrient broth, which would be in a test tube, and a nutrient agar, which is what I usually just refer to as a plate. It's also called a petri dish. Okay. So then before I even begin, I want to make sure, before I begin any type of transferring, I want to make sure that everything's properly labeled. So if I am using a broth culture, then I can use my grease pencil. Okay. If I've got a grease pencil, I can use my grease pencil to label the test tube, and there's usually a white spot on the test tube that you can use for a grease pencil. So you can only use grease pencils when labeling test tubes, because the grease can be cleaned off and the test tubes reused, right? For your plate, then you can use a grease pencil to label the plate. I prefer to use a Sharpie. Okay, it needs to be a fine tip or ultra fine tip Sharpie, and you label the plate around the perimeter of the back. Okay. So the plate is two halves. There's a lid. There's the lid of the plate, and then there's the bottom of the plate that contains the nutrient agar, right? So it's on the bottom of that, okay, where the nutrient agar half of the plate, there where you label the plate around the perimeter. All right, you never label the plate in the center, okay, of the plate. You never write in the center if you can avoid it, because then that makes it more difficult to take a look at your bacterial or your growth after incubation. So we can label it around the perimeter, and I'm just going to put some basic labeling here, like today's date, my name, what the organism is that I'm culturing, and there might be some other information required. And I'm going to put up a picture in this tutorial that will show you all that pertinent information that you need to put on your plate. But make sure that labeling comes first. Make sure you gather all your materials. Make sure everything's properly labeled, and then you're all set and ready to go. Okay, so one of the first things we need to do is set up a sterile field, and we need a heat source to work with to sterilize our inoculating loop, or whatever tools that we're going to be using to do the transfer. So the first thing we're going to do is, right now we're using Bunsen burners, so I'm going to look in my community drawer, right, and I'm going to find a Bunsen burner. Maybe the Bunsen burner is already out on the bench, and that would be okay too. I'm going to connect the Bunsen burner, the gas tube, to the gas line, right, and going to make sure that the Bunsen burner is actually screwed on properly, because these can get loosened up. The bottom portion of the Bunsen burner is meant to be adjustable, but sometimes these top portions, this top tube here gets loose. All right. If I look in my community drawer, I'm going to find some matches, right? I'm also going to find some other things, but for today, an inoculating loop. Okay, so here's my inoculating loop. So I'm going to need that inoculating loop in a minute, so I'm just going to set that aside. And for now, oh, one other thing that I need to pull out is I've got a little, there should be a little hot plate, okay, which is just a tile, right? The drawer that I just pulled out, it's missing. So if you are missing something, then you can let me know and we'll find what you need. This hot plate is used to, you can see it's got black marks, to put your matches on, or even maybe if you've sterilized your inoculating loop and it's hot, to put that on. All right. So the first thing we want to do is set up our flame. Okay. So this is where things get a little tricky. Now, but if you're going to work with a flame or live cultures or anything, one thing that I should have mentioned when we were talking about a tire is, if you're like me and you have long hair, then you can't do any of this until your hair is tied back, either in a bun or a ponytail, something, so that, and it really, if you lean over like this, however it's tied back, you need to make sure that it doesn't fall forward over your shoulders and make contact with the flame or bacteria or the bench or anything like that. So the hair needs to be tied back, because now we're going to be working with the flame. So the first thing I'm going to do is light my match. Sometimes it doesn't work out too good. So notice where I put the match. I put the match on the little hot plate here, okay? Never put a hot match or a recently lit match on the bench, because as you saw on this plate, it does leave a mark, okay? So let's try it again. All right. This one's working a little better. I'm going to hold it to the side, turn on my gas a bit, okay? You may not need to turn your gas on all the way, right? And you want to be very careful. You see how big this flame is? That hopefully it's coming through in the video. That is a pretty good sized flame, okay? That is a pretty good sized flame. So that's a little bit on the big side, right? So you have a couple of different ways where you might be able to turn down the flame. Some Bunsen burners have a little knob on the bottom and you can actually adjust the gas flow, all right? Some Bunsen burners, all Bunsen burners will have this adjustment here. So you want a nice blue flame, but you don't necessarily need a huge blowtorch. So I can turn down the gas some over here at the gas nozzle here, and you want your flame to be about 6 to 8 inches high, so this is about a good 6-inch flame. You don't need a big flame like this, okay? So a nice 6-inch flame or so. And the reason that you want a nice big flame, so now I'm done with these. I'm going to put them back in my drawer, okay? No need to have anything that clutters, okay? So when things are not needed anymore, best to put it away, all right? So the first technique I'll demonstrate is to do a subculture. So it transfers some bacterial cells from our actual stock culture or our starter culture. We're going to pretend that this is our starter culture, and we're going to transfer that or subculture some of those bacteria into our new fresh nutrient broth, okay? Our new fresh nutrient broth. All right, so first thing you need to do is to sterilize. So to do that transfer, by the way, you're going to use an inoculating loop. So you'll be using an inoculating loop a lot in lab in this class, okay? So I'm going to sterilize my inoculating loop. Never hold the loop choke up here, okay, because this will get hot, all right? So it's always best to be towards the opposite end, okay, to hold the loop towards the opposite end of the loop, like this, not like this, okay? So I'm going to hold the loop here, and I'm going to hold it at about a 45-degree angle, or maybe even a little bit more. So almost that entire, see, almost this entire, the loop and the wire are immersed in the flame until they're red hot. And usually if your flame is set a nice blue flame, it's only seconds that this takes. And once everything is red hot, okay, starting at the loop, move through to where the wire ends, and now that's sterilized. Okay, that's been sterilized. But it's hot. Okay? It's too hot. If it makes contact with bacteria right now, it's going to kill the bacteria because it's too hot. So what you need to do is sterilize and then cool, and it only takes about 10 seconds, right? And now I'm ready to grab some bacteria out of my culture here, okay? So this is my stock culture, right? And it's this test tube. It's got these slip-on caps. These are yellow. They might be different colors, all right? So I've sterilized, I've cooled my loop. Now I am going to use my pinky, right? I'm going to use my pinky. I'm going to pull the cap off, and I'm going to be somewhat close to my flame, right? So why am I close to my flame is because the flame is creating an updraft here. So if you stay in this, say, radius, this perimeter around your flame, then that's called a sterile field, right? So you want to work not too close, obviously, but somewhat close to your flame when you're working with live bacteria and when you're subculturing, all right? So I've sterilized, I've cooled. If it's been too long since I've sterilized, like I've been talking and everything for quite a while since I sterilized it, I'm going to sterilize it again, okay? And everything is going to get nice and red hot. I'm going to let it cool. Waving it around like this does not help it cool. You just need to kind of keep it near the flame because it's within the sterile field. Let it cool for about 10 seconds. Then I've got notice I'm right-handed, okay? So I've got my inoculating loop in my right hand. I've got my culture in my left hand. If you are left-handed, this would probably be the opposite, okay? I'm going to grab that cap with my pinky, okay? I'm going to pull it off, and I'm going to pass the top of the test tube through the flame twice. Never hold the test tube in the flame or it will heat up and break, and you might get a burn or a cut, or you might spill your bacterial culture, or nothing good will happen if you do that. So it's just a once, twice, like that. And then I'm going to go in with my inoculating loop and I'm going to grab some bacteria. Notice that I'm holding my test tube at about a 45-degree angle. That is so to minimize anything falling into the test tube, minimize the chances of anything falling into the test tube. Whereas if I hold it straight up like this, not only is it harder for me to work with, but then you increase the chance of something falling into the test tube and contaminating your culture. So you're always working at a 45-degree angle. Notice that I have not set down the cap. Never ever set down a cap on the bench, because then the cap might get contaminated. Then when the cap goes back on the test tube, then the culture might get contaminated, okay? I've got my cap, I've got my tube at a 45-degree angle, I've sterilized and cooled my loop. Now I'm going to carefully go down and dip the loop into the culture and then bring it right back out. You don't need to jiggle, you don't need to twirl, you just need to dip and pull it out. And then sterilize again, 2 passes. Put the top back on the test tube and then put the test tube back in the rack. And I've got 1/100th of a mil of culture, of that bacterial culture, lots of bacteria in the loop part of this inoculating loop. And if you look closely, you can actually see the liquid, right? So there's where my bacteria are that I'm going to transfer to subculture into my fresh nutrient broth, right? So you're going to do the same process with the nutrient broth. Pull the cap off with the pinky, 2 passes in the flame. I'm going to go down with my inoculating loop. You don't need to jiggle, you don't need to twirl. Go down, pull it out, 2 passes, recap, set to the side, and now sterilize the loop once again. Okay, now the loop's nice and hot. I may want to put it down, so I can put it down on my hot plate and then I'm either done or I'm ready to inoculate something else or subculture onto another medium, right? So with the inoculating loop, you sterilize it before you begin and then when you're done at a minimum, all right? Depending on what else you're doing, which is I'm going to show you next, you may have to sterilize the loop more times than that. So that was doing a subculture from a liquid culture into a nutrient broth. Now let me demonstrate how to subculture from a nutrient or from a liquid culture onto a plate on a petri dish, right? So this is a solid medium, and I'm going to demonstrate the single colony isolation method, which is a common method for getting single colonies of bacteria and for diluting out bacteria. So even if I have multiple different strains or species of bacteria in this tube, if I do the spread plate technique, the single colony isolation technique properly, I'll be able to isolate those individual colonies and make pure culture. So this is a way to isolate pure colonies of bacteria. And we'll talk about what colonies are and how they come about in lecture and lab. This, again, we're going to focus on the technique, right? The technique part. So there's a four-quadrant. This is a four-quadrant streak method. So I'll put up a picture of what I'm talking about in terms of the quadrants, and there'll be information available on Canvas as well. So the first thing is first, okay, I've got my petri dish labeled already, right? So I'm good there. I'm going to sterilize the loop first, as always. Make sure you sterilize it all the way, the wire all the way up to the handle. I'm going to let it cool for a few seconds. I'm going to grab my culture, my bacterial culture, in the test tube. I'm going to pull the cap off, 2 passes in the flame, hold at a 45-degree angle, go down, pull out some bacteria, 2 passes in the flame, recap, set this aside. And now I'm done with that culture for this technique, right? I will not be getting any more bacteria out of there to spread on this plate. Once and once only to do quadrant 1. Bacteria is here. I'm still close to my flame. Petri dish is upside down, agar side up. The reason I do this is you grab only the agar side and you leave the lid sitting on the bench. Grab it, flip it over, and then I will spread out quadrant 1, several passes back and forth in that one section and one section only, which is the first quadrant of the dish, of the plate. And then I'm going to flip it back down, right? I'm going to rotate a little bit, and then the next time I pick it up, I'm going to streak out quadrant 2. But before I do that, I've got to sterilize my loop. So you sterilize and cool your loop between each quadrant, but you do not get any more bacteria out of the original culture. So this is a way to dilute out the amount of bacteria that there is in each quadrant on the plate. So I've cooled, I've sterilized, I've cooled. I'm going to flip it over. If you kind of angle it just right, you'll be able to see your first quadrant. There's no growth there yet. The bacteria that are there are invisible, but if you angle it just right, you'll be able to see it. So I'm going to make sure that I streak out my second quadrant. Put it back down on the lid, sterilize again, cool, rotate my plate a little bit. Again, I'm not going back into my original liquid culture, right? Now I'm going to streak out quadrant 3 on the plate. This is cool, sterilized and cooled. I'm going to angle so I can see quadrant 2 and then streak out quadrant 3. Put that back down, okay. Sterilize, cool, right? rotate, and then the fourth and final quadrant. This is the one that you have to be most careful about, because when you're streaking out your quadrants, you want to be careful not to overlap older quadrants, okay? So now I'm going to streak out quadrant 4, and I'm done. One final thing to do, that is sterilize the loop. All right, you can, once you get more comfortable with working with the Bunsen burner and your loop and sterilization and everything, then you can even increase your angle and get more of the loop in there at the same time. You see that? You can really sterilize the loop really well. If you do that, you've got to be really careful not to go too far and burn your finger up here, because if I were to go too far and then put this down in there, I would burn my finger. So you've got to be careful. You can increase your angle there to get a nice red hot loop, okay? But I will warn you that if you hear this, so before I mention that, I want to say we're done with this, right? So now I've sterilized. I can then put this down to cool, and then I can put my broth and my plate wherever they need to go, in the incubator and in the tray in the front of the class to be incubated, in the refrigerator. Depends on the situation, okay? But I'm done with these, and once my loop cools, then I can put it back in my drawer. You want to make sure that it cools first, right? You don't want to stick it in your drawer hot, okay? Right? You don't want to stick it in there hot. So make sure it's cooled. I'm going to pull it back out. All right. I want to demonstrate very quickly what can happen if you do something wrong. All right. So now, let's just say that I'm going to go through something here, and hopefully you can identify what I'm doing wrong. Okay. Right, that is some really poor aseptic technique right there. That would equate to certainly point deductions on your lab technique, which would show up as point deductions on your lab report. Again, if you're going to be in this microbiology lab, you have to learn proper, and you're going to be working with live microbes, live bacteria. You have to learn proper aseptic technique and adhere to those protocols. Again, attention to detail. So I did a lot of things wrong there. I didn't sterilize when I was finished. I didn't put my loop in. I didn't properly sterilize my loop, so my cultures are probably contaminated. I didn't put the cap. I threw the cap on the bench here. I didn't put the cap back on. Lots of different things that I did wrong there. All right. So again, when you're doing this, you're going to sterilize. Make sure you sterilize the whole wire, but certainly the loop that you cool. Oh, another thing I didn't do is I didn't sterilize the test tube, the top of the test tube. Okay, I held my test tube up straight instead of at a 45-degree angle. Okay, I didn't re-sterilize my test tube afterward, and I didn't cap it. Right, and then when I was all done, I didn't sterilize my loop and wire as the final thing, right? My final step. Here's another thing that another mistake that's fairly common. You sterilize that loop nice and hot. You get the whole thing in there. You get it all nice and hot. You did a great job sterilizing, and then listen. You may not have heard that, so let me try it one more time. You got to listen for it. That sizzle, if you get a sizzle when you stick your inoculating loop in your broth, or you get a sizzle when you are, see that? You get a sizzle, or it literally melts the agar when you stick your loop in the broth, or when you stick your loop on the agar, you have not properly cooled your loop, and you have just possibly killed all your bacteria, right? So make sure that you properly cool your loop. When you're working in a broth, you just have to be patient and just let it cool in the air, right? If you're like me and you're less patient when you're working with agar, here is a technique that you can use to rapid cool using, if you're using a plate, right, an agar plate, and that is, you do your sterilizing, but instead of waiting in the air, you can stick it on the side, on the very edge of the plate, and it'll sizzle, and then it'll melt into the agar, but now it's cool and it's still sterile, so then I can do my streaking or whatever it is I'm doing. Okay, so you can rapid cool when you're working with a plate, because you can stick it in the agar. Just make sure you stick it in on the very edge. Don't stick it in the middle, you know, or where your bacteria is or something. All right. And then, of course, again, when you're done with everything, you're going to properly sterilize. You're going to sit there and let it cool, right? You want to be very careful when you don't need your flame anymore. Okay, you're done working with the bacteria. Everything is properly capped, and the petri dish is, of course, got the lid on it and so forth. Now you can carefully turn your flame off. Always have situational awareness where your flame is. Obviously, you don't want to reach over your flame, okay, or you don't want to stick your, you don't want to reach over this way and then stick your face too close to the flame. Okay, so be very careful. Make sure you have a high degree of situational awareness when you're in the lab, especially when you're working with the Bunsen burner and an open flame and live cultures. Okay, so I'm going to carefully reach over, turn that off. Okay, now this is a bit on the warm side here at the top. Okay, so I'm going to let that cool for a little bit too. It doesn't take very long for these things to cool, a minute or two, and then you can put them away, right? So now this is nice and cool. My inoculating loop, I can put it away. Okay, matches don't go in here. Okay, use matches don't go in here. Use matches don't go over here in the sink. Use matches go in the trash, right? So make sure when you're cleaning up that you put your matches in the trash. Okay, this goes back in the drawer, right? And again, and the Bunsen burner can go back in the drawer unless I've told you to leave them out. Then when it's cooled, again, it's only been a couple of minutes, but it's plenty cool. Carefully put that back in the drawer. And now your plate goes wherever it needs to go. Of course, I'll have told you where you need to put it. Your broth goes wherever it needs to go. And then when you're done with your cultures, if I've told you that you're to dispose, anytime you dispose of any live organisms, they don't go down the sink. They don't go down the trash or in the trash. There is a biohazard disposal area, right? So I'll show you where that biohazard disposal area is. It's over in the corner of the lab, the far corner over there, right? And let's just walk over there. Okay, so I'm going to take my my used starter culture over here to the biohazard area. It's labeled biohazard. Cultures would go in the test tube rack. Plates would go in the plate rack, right? So these would never go in the trash, right? Cultures would never be dumped down the sink, okay? Definitely properly disposed of. They get sterilized before they are then properly disposed of. Okay, so that's it.